Hello everyone, this is Science Vines and you are watching the audiovisual chapter of class 11th biology. This is the third part of chapter 6 anatomy of flowering plants. We are starting off with the dicotyledonous stem. The transverse section of a typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem. Figure 6.7a. Covered with a thin layer of a cuticle, it may bear trichomes and few stomata. The cells arranged in the multiple layers between epidermis and pericycle constitute the cortex. It consists of three subzones. The outer hypodermis consists of a few layers of collenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Then, cortical layers below hypodermis consist of a rounded, thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grains, and the layer is also referred to as starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are a few layers of radially placed parenchymatous cells, which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundle is a characteristic of a dicot stem. Each vascular bundle is conjoint, open, and with end-dark protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces, which occupy the central portion of the stem, constitute the pith. Now, come to the monocotyledonous stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Figure 6.7b. Let us look at the figures as well after we complete this. Now, vascular bundles are conjoint and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water-containing cavities are present within the vascular bundles. Now let us look at the diagrams. Let us first look at the transverse section of a dicot stem. The diagram A, or the first diagram showing the epidermal hair, then epidermis. Under the epidermis lies hypodermis, then the parenchyma cells, then comes the endodermis, and then the pericycle, and then there are vascular bundles, and in between the vascular bundles there are medullary rays, and then comes the pith. Magnified view is present on the right side, which is showing epidermis, and epidermis is actually covered with a thin layer of a cuticle, and it may bear trichomes and few stomata as well. Then comes the hypodermis, which is by collenchyma cells. After that, there comes parenchyma cells, which are actually the cortical region. Then comes the endodermis, which is rich in starch and often called as starch sheath. Then comes the pericycle, which is present in the form of semilunar patches of sclerenchyma. Then comes the vascular bundles, which are conjoint, open, and with end-dark protoxylem, which means the phloem and xylem are joined together, and the end-dark protoxylem means the protoxylem is inside towards the pith, and open type means a cambium is between the phloem and the protoxylem. Then comes the pith. Pith is large number of rounded parenchyma cells with large intercellular spaces. So here we end this transverse section of a dicot stem. Now let's see the diagram of a TS of a monocot stem. And it is showing the epidermis, hypodermis, vascular bundles and the ground tissue. Let's elaborate a bit. So the epidermis after hypodermis comes. The hypodermis is of sclerenchymatous tissue, and then comes the scattered vascular bundles, actually in a large number as well. Each is surrounded by sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Here vascular bundles are conjoined but closed, okay, which means there is no cambium in between the phloem and the xylem. Then peripheral vascular bundles are smaller than the centrally located ones, and phloem parenchyma is absent, and the water containing cavity is present within the vascular bundles. So this is all about the transverse section of a monocot stem. Now let's talk about the dorsiventral dicotyledonous leaf. The vertical section of a dorsiventral leaf through the lamina shows three main parts, namely epidermis, mesophile and vascular system. Epidermis, which covers both the upper surface, adaxial epidermis, and lower surface, abaxial epidermis of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. Tissue between the upper and the lower epidermis is called the mesophile. Mesophile, which possesses chloroplast and carry out photosynthesis, made up of parenchyma. It has two types of cells, palisade parenchyma and the spongy parenchyma. Adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells, extends to lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles, which can be seen in the veins and midrib. The size of vascular bundles are dependent on the size of veins. Veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Look at figure 6.a and find the position of a xylem in the vascular bundle. Now let's talk about the isobilateral or monocotyledonous leaf. The anatomy of an isobilateral leaf is similar to that of a dorsiventral leaf in many ways. It shows the following characteristic differences in an isobilateral leaf. The stomata are present on both the surfaces, and the mesophile is not differentiated into palisade and a spongy parenchyma. In grasses, certain adaxial epidermal cells along the veins modify themselves into large empty colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, They make the leaves curl inwards to minimize the water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles, except in main veins, and seen in vertical sections of leaves. Here we end the third part of anatomy of flowering plants. To watch more such videos, do subscribe the channel and press the bell icon so you don't miss any video. And if you want us to create more such videos on any other book or subject, do mention in the comment section please. Like and share the video to keep us motivated to create such content. Thank you for watching the video. Bye bye. Love.